Okay, this is it. This is the last video in the biopsychology series. We're going to be looking at biological rhythms. It's another quite tough one, so maybe watch it twice. Be quite careful going through it. This is how I've set out the page. You'll want to do something similar if you're using the mind map. If you're just using A4 pieces of paper, just make sure you get all the notes down, and we'll work through each of the biological rhythms in series. Okay, so first of all, we're going to look at what two new terms are, and we need to add them to your vocabulary. What we have is we have endogenous pacemakers. Now, endo means kind of inside, and then the pacemaker is like a clock, so it's our inside clock, it's our internal body clocks, and these regulate our biological rhythms. These keep our biological rhythms to time. So these are already inside us, keeping the circadian, infradian, ultradian rhythms in sync effectively. We also need to know about exogenous, or exo, like outside, like an exoskeleton is an outside skeleton. So maybe more confusing than I need to be. Zeitgebers. So this is a external cue, which is going to switch your internal body clock to a new cycle. It's going to manipulate your internal body clock. So we need to know about the role of endogenous pacemakers and exogenous zeitgebers a little bit as we go through each of them, but we really need to know their roles in the circadian cycle for this section. Okay, so the circadian cycle is, well, circa means about or around. Dian means a day, so we'll infradian, ultradian. So they're all talking about time in relationship to the day. So circadian means about a day, and the most obvious cycle that we go through over the course of 24 hours or so is our sleep-wake cycle. So we sleep, and we go and we wake up, and then later on we need to sleep again. Now we might think that the sleep-wake cycle is just a habit or just a cultural aspect, and we can, with effort, stay awake for relatively long periods of time, but generally we do have a regular 24-hour sleep cycle. Now, to test this scientifically, we have Sefer, I guess his name is, and we can see him on the right hand side. This is back in 1972, and he went to Texas, got underground, was plugged up by NASA, who monitored his bodily processes, and he spent 205 days in this cave. So the better part of a year sitting in this cave. Now that must have been an incredible chance to catch up on his reading. But importantly for our interests is he didn't have any kind of external exogenous zeitgebers that kept him to time. He had no watches, he had no natural light. He had artificial light, but he didn't know when to turn it on and when to turn it off in order to keep him on a regular cycle. He did keep his own regular cycle. He did feel sleepy at the same time and spend the same time awake, same amount of time asleep. Pretty broadly similar to someone's general sleep-wake cycle. He did have a sleep-wake cycle just north of 24 hours. So it was longer than 24 hours, and you do see that across a lot of sleep research, that actually the natural cycle is slightly longer, which can mean if that just was all you had to go on, you would drift out of sync with the day-night cycle, which is exactly 24 hours. And Srifer did find this when he left, and actually he realized that, well, 205 days have gone by. He didn't feel like 205 for him. He was keeping a record, and he found more time had gone past than he expected, and it was quite a depressing thing for him to realize that more of his life had passed than he expected. Okay, so a couple of evaluations to see if this is what we see. Ashcroft and Weaver did a very similar study, but they used groups instead of an individual. They isolated these groups inside World War II bunkers with no natural light, just artificial light. And these individuals reported a 25-hour cycle, so again, slightly more than the 24-hour cycle that we expect. And we will see another evaluation in a second about someone else with a longer than 24-hour cycle. So it can be useful for us to know about these biological cycles, especially the circadian cycle, because part of this sleep-wake mechanism is also a process of different bodily activities peaking and dipping throughout the night. So blood pressure, digestion, all that kind of stuff will vary through a night. So one application is when to take aspirin. If you take aspirin about 11 o'clock at night, it'll peak at about 3 or 4 in the morning, which is the kind of time that people tend to have heart attacks. And aspirin is quite useful for reducing the chance of heart attack. Also, if we can figure out more about the sleep-wake cycle, we might be able to counter the effects of jet lag or shift work. Okay, so we do need to focus particularly on endogenous pacemakers and exogenous indicators in relation to this sleep-wake cycle. We have identified the endogenous pacemaker for the sleep-wake cycle. We know exactly where it is and where it is. It's been identified as the suprachiasmatic nucleus, not an easy word to say or spell. And it's within the hypothalamus, so it's deep within the brain. Few bits of research to find this out. One of the most interesting ones is there were some abnormal hamsters. And these hamsters had a sleep-wake cycle of only 20 hours. So they were, most creatures have a sleep-wake cycle of around 24 hours because it ties in with the light around of the earth. So if you don't want to get eaten, you're probably going to want to go get your food during the day and then maybe sleep at night. But these hamsters had a natural 24-hour cycle, so there was something going on with the suprachiasmatic nucleus. They had their suprachiasmatic nucleus removed and placed inside rats who used to have a normal 24-hour cycle. Again, with the rats' brains. Well, rats. These rat cycles then change from 24 hours to 20 hours. So it certainly was something about the suprachiasmatic nucleus that was causing the internal sleep cycles of these hamsters. No hamsters survived. Okay, so that was the endogenous pacemaker. We're fairly confident that the strongest exogenous zeitgeber is sunlight or natural light, and the body clock will adapt to exposure to natural light. One example is someone who never had exposure to natural light. So Miles reported on a case study of a blind man who had a natural 25 effectively hour cycle. Now, if you've got a 25-hour cycle and you can't see light to reset your endogenous pacemaker, you're very quickly going to go out of sync. So you're going to wake up maybe an hour later every day to everybody else. And over the course of a month, you will go through a whole 24 hours of kind of shifting later and later and later. So at some point, you'll be waking up feeling you've had a good night's sleep, and it'll be 2 in the morning, and everyone around you is still asleep. So what this individual did is he had to take sedatives and stimulants to try and maintain himself on a cycle similar to everybody else, but he found it incredibly hard. Social cues, like when everyone else was around and awake, alarm clocks just really didn't help in him trying to maintain a normal cycle. Okay, so we're going to talk about infradian rhythm now. This is less than once a day. So it could be up to a year, it could be an annual cycle, it could be a weekly cycle, it could be, as we'll look at here, around about a monthly cycle, because we're going to look at the menstrual cycle, which seems to be the clearest cycle. So the menstrual cycle is a regular 28, it can vary, but around about 28-day cycle of fertility. So McIntosh did a classic study on this. It was a 10-year longitudinal study involving 29 women. They were around 20 to 39 years old, and 20 of them were the sniffers, and 9 of them were the donors. So the 9 donors gave sample pheromones. So the pheromones are acting as the exogenous Zeitgeber here. These are influence and affecting the endogenous pacemaker, which is keeping that 28-day cycle of fertility. So these 9, well, I prefer to be one of these 9 and the other 20. These 9 participants would keep sweat pads under their armpits, and over the time the pheromones would secrete into these cotton pads. They would take them from under their armpits each day, put them in a little test tube and post them out. These would then go to women at different times in their menstrual cycle. They would wipe the pads under their nose each day, and over the course of this study, over the course of the 10 years, 68% of the women who waved their pads under their nose responded to these chemical signals, and their cycles switched to match the ladies who had sent the pheromone cycles. So yeah, big gross. So about some evaluations on this. We could argue that McIntosh study is quite artificial. I mean, that has strengths. The fact that these women were separated from each other and didn't know each other's identity maybe takes some of the potential issues away. But Russell did a similar study looking at a group of women who lived together at universities, and they didn't take oral contraception, which obviously can affect the menstrual cycle. Found that these groups of women who lived together, very close quarters, would sink over the course of their time living together. However, another piece of research that looked at a women's basketball team, they travelled around America together for extended periods of time, quite a lot of pheromones because of the amount of sweat that would have been produced under these circumstances, but there was no evidence of synchronization. I've never been a massive fan of this study, mostly because the idea, I don't know how many people would be on a basketball team in total and traveling around together, but I imagine with the sheer number of pheromones of all people at different stages in the cycle, it would be hard to kind of lock onto one to synchronize with. But one of the criticisms of the infradian cycle is we're not really sure why women would link their menstrual cycles together. It would be, of course, an evolutionary reason, and I do have a potential idea, but I'll be interested to see what you can think of before the class. Okay. The final cycle is the infradian cycle. So these are cycles that are more than once a day. So you'll have many of these over the course of a day, well, actually over the course of a night, because we're going to be looking at the stages of sleep. So when you go to sleep, it's not just one solid process where you are asleep and that's it. You will have a range of brainwave patterns which represent different stages of sleep. And you will move through these throughout the night, and these stages will repeat. So the original research, Dermot and Kleitman, was using an EEG, and this is when EEGs were fairly new. They would use these EEGs to investigate brain activity. So to fall asleep, EEG hat goes on, and they would watch you in a sleep lab. All very, very normal. 9 participants took part in this. They hadn't drunk caffeine, they hadn't had any alcohol, and they went to sleep in the sleep lab. So they would watch the brainwave patterns, and they would also wake them up at different stages as well. If you woke them up in REM sleep, something interesting happened, they would report dreams, giving us an indication of the function of REM sleep. And in fact, they could see how the EEG readings would change depending on how vivid the dream was. So if you woke someone up during quite an intense dream, you could probably predict that on the basis of the EEG. Okay, so below we have a range of EEG signals, and I also have a visual representation of the kind of staircase of sleep. So we can see the diagram on the bottom left. From the top, which is bright green and awake, we will drop down to stage 1. So you'll slip, you will go straight past REM. You won't go into REM straight away. You go into stage 1, then down to stage 2, then down to stage 3, then down to stage 4, which is incredibly deep sleep, then back up and then into REM, and then you'll spend some time in REM, and then the cycle will repeat to stage 1. So let's have a closer look at some of those stages. So you're awake, brainwaves are normal, you're active, you're going through the day. Then you start to fall asleep kind of at stage 1. Stage 1 isn't even quite asleep. 5 to 15 minutes, you'll be very, very easy to wake, but you're just more relaxed than you usually would. Your brainwave patterns are very similar. Your heart rate will start to slow down, but that's about it. We get to stage 2, you are asleep now, and we can start to see these small high frequency bits on your EEG, which is called a sleep spindle. The body is much more relaxed now, and you are harder to wake. You'll go into deeper sleep, and then you're going to start to get some delta waves, so some of the bigger waves on the brain, so some of the kind of high amplitude waves. Spend about 15 minutes in stage 3. You're very relaxed and you're much harder to wake than you were in the previous stages, and then you're going to slip into stage 4, at which stage we get what are called delta waves, which are the slowest waves. So your brain isn't working too fast on this, and we're spending a significant amount of time in stage 4, so about 40 minutes. At this stage in the body, growth hormones are going to be released into the bloodstream, and it's generally thought this is a stage of sleep in which your body is repaired. Then, after a certain amount of time in stage 4, you're going to go quickly back up through these stages. So you can go back up to stage 3, then back up to stage 2. Then you're going to go straight from stage 2 into REM sleep. So REM stands for rapid eye movement. So this is, if you open someone's eye at this stage, you could see their eyes moving about very quickly. You're going to have the most brain activity in REM sleep, very similar to waking brain activity. And of course, as we said, if you're woken up at this stage, you're going to report dreams. Also, your breathing rate becomes regular as well as your heart rate as well. After a certain amount of time in REM, about 15 minutes, you're going to go back to stage 1, and you're going to repeat that for 90 minutes. Some of the stages will change slightly in length, but we're looking at around 90 minutes, and then that's repeated a number of times, and then you'll wake up after a night's sleep. So let's have a little look at some evaluations. Okay, so linking onto the stages of sleep having functions, and as well the role of exogenous light gavers, Sharpo looked at ultramarathon runners, so people who really, really push their body. After ultramarathon runners had finished their exercises, they would sleep for much longer than normal, which is kind of to be expected. But when their brainwaves are measured on EEG, they found that 45% of the time was spent in stage 4. And considering their body was pretty broke down, this does suggest that stage 4 is for bodily recovery. Research has shown as well that patients who've trying to recover from drug overdoses or brain damage will spend longer in REM sleep. So this would suggest that REM sleep is there for the recovery of the mind. Now these results, these stages, these brain stages, have been found in humans, but they've also been found in many animals as well. So we're quite confident that these are fairly standard stages of sleep that are common to most animals. Okay, so you've just seen some of those sections zoomed in a bit if it was hard to read, and that is it. The mind map is complete. That should be all you need to know about biopsychology, which is generally seen as the hardest, if not one of the hardest units in psychology. So keep that mind map close to hand for your revision, and keep coming back to it to keep the information fresh. Obviously, you want to see a full set of notes, so either the mind map or if you are working on A4, the same amount of detail on A4 paper. When you come in, we want to see if we can apply some of these cycles to different scenarios, and of course, hopefully you're going to be ready for a quiz when you walk in.